Hello, you are listening to 1 in 2, a Manchester Cancer Research podcast, brought to you by the University of Manchester and the Manchester Cancer Research Centre. With 1 in 2 of us receiving a cancer diagnosis at some point in our lifetime, it has never been more important for our research to improve the outcomes of people affected by cancer. I am your host, Sally Best, and throughout this series I will be speaking with Manchester cancer researchers about their innovations, discoveries and projects that are changing the landscape of lung cancer detection and treatment. Lung cancer is the leading cause of cancer death worldwide, causing 35,000 deaths in the UK each year alone. As such, it is one of CRUK's 4 cancers of unmet need. Of 47,000 new lung cancer cases every year in the UK, 15% of those diagnosed are never smokers. Here in Manchester, we have one of the worst lung cancer mortality rates in England, so we are striving to improve early detection and treatment of this cancer. Across these episodes, we explore lung cancer from the genetics and diagnoses to screening and treatment, talking to Manchester researchers who specialise in basic, translational and clinical lung cancer research. And remember, you only need lungs to get lung cancer. In this episode, we speak to Professor Caroline Dive about cancer biomarkers, including liquid biopsies, which involve sampling a patient's blood to determine the presence of cancer cells or cancer DNA. We discuss the Cancer Biomarker Centre here in Manchester and the work that Caroline and the team are doing to develop lung cancer biomarkers. We go on to cover how Caroline sees the future of integrating novel biomarkers into the clinic for their use in a routine setting. Hello, Caroline Dive! We've got you here, finally.
Very good. Well, it's a pleasure to be here, Sally.
I'm so glad. So we've got another episode of Season 2 today. Season 2 is focusing on lung cancer. So we're going everything from the basic biology through to treatment, diagnosis, and then kind of to the pathway, which will be some of our later episodes. But we've got you on today to talk about, I think it's quite a complex subject. So we will start at the beginning, I think is obviously always the best place to start. But you're very good at talking about this. So I am so very sure we are in safe hands for this. So, I mean, how are you doing, first of all?
Oh, we're thrilled because, we've just moved from Alderley Park.
Yeah.
The fire to the Patterson Institute was in 2017. So we've been in exile in Cheshire, in wonderful accommodation, I have to say, in Alderley Park, but not close to the Christie Hospital. Yeah. So we've just moved back into the most extraordinary research facility in the Christie Hospital site with all our colleagues who do cancer research from the faculty. It's brilliant to be there. It's brilliant to be back. And all the sort of collaborative research that I do is done best right where we are now.
Yeah, it's fab. So for those of you that don't know, the Patterson burnt down, as Caroline said, in 2017. And there is now 150 mil build, and it's incredible. It's right on Wilmslow Road, and I got a little tour of the Biomarker Centre, which we'll talk about, and it was incredible. I was like an energised puppy looking around all the labs. It's a great space, and like you say, there are so many different teams in there because it's got the CRUK MI, some of the Christie, some of the MCRC, some of the uni, so it's just this massive collaborative space, which we'll come on to talk about the benefits of that and the benefits of that close proximity research. But I mean, just to get started, are you okay to tell our listeners a little bit about you, what you do, your responsibilities?
Sure. I mean, I started my career as a pharmacist. I trained in London in pharmacy. I then decided, after being fully trained, that actually I wanted to do cancer research. And the sort of motivation for that is personal, as it is with many people. Cancer touches most families' lives sooner or later. For my family, my maternal grandfather died of a brain cancer just a few weeks before I was born. Had a devastating impact on my mother. And I did think at the time when I just finished my pharmacy training that I actually wanted to go and do something about it. So I went into cancer research, did my PhD in Cambridge with Paul Workman, really working on drug resistance, cancer drug resistance. And then I took a career path which was a little bit unconventional. I completely missed the postdoc piece and went from being a PhD student to a principal investigator. That was very stressful.
I can imagine.
Sink or swim. I decided perhaps I should swim. I studied apoptosis, or programmed cell death as it was then called. And that was quite a new subject area at the time. So I spent more than a decade doing really fundamental curiosity-based research. Moved to Manchester 1990, I think. Continued to do that for some time.
Yeah.
And then Nick Jones, who was then the director of the then Patterson Institute here at the Christie Hospital site, asked me if I would like to join the institute. So I stopped being a division chair in what was then the Division of Pharmacology, Physiology and Toxicology, if I've got that the right way around.
They change all the time.
Completely restructured, of course, the faculty now. And I moved over to the Institute. And one of the things that Nick wanted me to do was to work with the phase 1 trials unit at the Christie Hospital. He knew I'd been a pharmacist in the past. He knew that I was used to working with clinicians, that I'd done that in my training. And he wanted to work on a Friday afternoon to do pharmacokinetics, to work with the clinical trialists to find out how long drugs stayed in bodies and whether they worked, and do that kind of research. So I came with a basic science research program, but gradually realised, and it didn't take long to realise, that once I was here, the Patterson Institute on the Christie Hospital site, personalised cancer treatment was just coming over the horizon at that time. Back then in the early, late 90s, early 2000s. And we didn't really have what I would call that much biomarker research going on in the Institute. So there was incredible opportunity to build a cancer biomarker centre. Yeah. And that really started to get going in about 2005. Okay. So I've been doing that for quite a long time. It's been my life's work pretty much, and we are where we are today in a brand new cancer biomarker centre in the new building. Yeah. So that's a quick tour of how I got to where I am now.
Quick tour. I love that. A little synopsis of Caroline Dive's life. And I mean, did you plan on staying here this long in Manchester?
No. I come from Sussex, and it's tough up north, isn't it? So I didn't. I thought I would come to Manchester for 3 years, and 3 years has become. Well, I came in 1990, so do the maths. That's a lot of years. And I think, I love it here. I mean, it's the best place to do what I do. I truly believe that in the country. I think to do the work that I do, the University of Manchester, its medical school, and the Christie Hospital with the Cancer Research UK Manchester Institute, you put all that together, and it's the very best place for me to do what I do. And that's why I'm still up here in the north.
And you're not leaving.
I still can't have gravy with chips. Just can't do it.
Oh, God, Caroline, come on. You can. And cheese on top.
No, really
it's gotta be. Oh, nightmare. Well, so you've mentioned the Cancer Biomarker Centre. Can you tell me a little bit more about that? And also maybe kind of a brief overview of what exactly a biomarker is for those people that aren't too sure on that.
It's pretty much a catch-all term. And I think most people would think about biomarkers, when they think about that, they would think about it's just a test. Yeah. So it's a molecule or molecules measured objectively, usually in a clinical sample, but it could be in a preclinical model, that tell us a little bit about whether a patient is healthy or has a disease. It doesn't have to be cancer. There are biomarkers for all sorts of diseases. It tells us how that patient might do, their prognosis. It may tell us what type of therapy they will respond to. It might tell us whether they're at risk of a toxic side effect. It might help us diagnose cancer early detection biomarkers. So biomarkers is a whole range of things, but essentially molecules or molecule measured in a clinical sample usually that help us support the management of a patient.
Okay. So go on, tell us about the CBC.
So the CBC has just grown and grown. So when I first arrived, and I was supposed to be doing that pharmacokinetics with the phase 1 trial unit on a Friday afternoon, it did become clear very quickly that there was this massive opportunity to develop cancer biomarker sciences here in Manchester. We started by thinking we would get tumour biopsies, bits of patient's tumour to study, to try and understand the biology of each patient's tumour, and then to try and figure out perhaps what the best drug would be, for example, one of many things we thought we would be able to do. And actually it became very clear quite quickly that we didn't get tumour biopsies very often. Yeah. So then that sort of pragmatically led us to start thinking about measuring biomarkers or tests in blood. Yeah. And everyone's heard of prostate-specific antigen or PSA. Yeah. That's a classic example of a blood-based biomarker. We now call them liquid biopsies. Yeah. So not tissue biopsies, but liquid biopsies. And this whole field has just exploded in cancer research. And I think, it's been a long time coming, but we were one of the earliest groups that set up labs to develop liquid biopsies to predict responses, to measure responses. Is a patient responding or not?
Yeah.
And difficult to do that if you don't get not just one biopsy at the beginning, a tumour biopsy, but then another one as they go through their treatment.
Yeah.
But of course you can much more easily get over and over a sample of their blood to study. Yeah. So that's what we've done a lot since the beginning of the Biomarker Centre. We've tried to develop an expertise in this liquid biopsy approach. And the two major types of measurement we make in blood are circulating tumour cells or circulating free tumour DNA. Okay. So this is DNA that's been shed from a patient's tumour as a result of the proliferation and cell death that goes on inside tumours. That DNA is shed into the bloodstream where we can find it. It's tricky because we have to make sure it's tumour DNA. Yeah. And it's still a very vanishingly small amount of the DNA that's in the blood. So we have to have sensitive methods to find it and measure it. Mm-hmm. And that can tell us what type of mutations a particular patient's cancer has. And that in turn can help us determine what type of therapy might be best for that patient.
Okay. So you've said about this circulating free tumour DNA. So why is there a higher level of that in the blood? Why do tumours release that DNA into the bloodstream? Is it known or?
Well, it's a good question. It's not precisely known. Yeah. And I think a lot more research is needed to find out more about how and why tumour DNA is shed into the bloodstream. It's thought that it happens when tumour cells die. Okay. But the amount of tumour growth is a balance between how rapidly the tumour is proliferating and how many of those tumour cells are dying. And I think both proliferation and death and the balance of those two are probably important parameters for how much circulating tumour DNA there is in a patient's bloodstream. And it varies depending on what type of tumour they have. Okay. So some tumours are good at shedding circulating tumour DNA into the blood, and other tumours less good. Okay. So it may or may not be the answer to everybody's tumour, but I think for many tumour types now, all over the world, people are measuring circulating tumour DNA to monitor how well a patient's responding to therapy. So do the levels drop if the therapy is working? Do the levels come back up again if the tumour becomes resistant and it starts to come back? So on a simplest level, elements of circulating tumour DNA measurement can just tell us perhaps a reflection of how much tumour burden a patient is carrying. Yeah. But also by sequencing that DNA, it can tell us, well, these were the mutations they had at the beginning, and that's why we gave them a certain type of targeted therapy. Mm. But now, as they become resistant, there are different mutations potentially. Yeah. So it's a way of tracking how a tumour changes over time through therapy.
And I can imagine that's very, very useful to know in terms of the specifics of the treatment that you're allocating. And we'll come on to lung cancer, but
there's lots more. It's even more exciting than that because it might tell us when to stop treating with one drug and start treating with another one. So, there are lots of ways now that these liquid biopsies are being used. The one that's very much in the public eye at the moment is GRAIL and a liquid biopsy for early detection of cancer. Yeah. And I think we all sign up to I'm sure it's completely true that if we find cancer early, we have a much better chance of that patient seeing cure. Yeah. So that's probably one of the most important utilities and developments in the liquid biopsy field.
For sure. And I've just got a couple more questions about liquid biopsies in particular. So I'm just wondering, what is the sample size that you'd have to take? Does that adjust? And how long does it take to process that sample? Because if a patient comes in, and I guess it's kind of beneficial if it's processed within a shorter space of time so that treatment can alter or whatever within that short space of time. I don't know the specifics.
Yeah, the field is maturing. So a lot of the research that goes on with liquid biopsy is in clinical trial activities to actually try and test to see whether this new liquid biopsy is going to be useful in routine use in the NHS, for example, in this country. So we have to do an enormous amount of experiments in a lot of patients to be sure the test does what it says on the tin. Yeah. We can't just use, a lot of the work is in research mode. And, a lot of biotech companies are also developing liquid biopsies, and we work with some of them. And the idea is to do enough testing of the new test to make sure that new test is going to be clinically useful. Yeah. So for the last, in the early detection space, the last thing you'd want to do is tell someone they have a cancer when actually they don't, and your test is not accurate. Yeah. So there's a lot of work that needs to go in. It's called validation. You have to set up big, well-designed clinical trials to actually prove that a liquid biopsy does what you want it to do, accurately, robustly, reliably, reproducibly. Ground truth.
Yeah, exactly. A lot of work. And just going back to your research area of expertise, so you're in small cell lung cancer, which is around, if I'm right, 10 to 15% of the lung cancer population. And is it the most aggressive type?
It is. I mean, the Biomarker Centre works across all tumour types, but part of my research group is very focused on small cell lung cancer.
Okay.
And we got to that almost by some sort of lucky happen chance, because I work very closely with Professor Fiona Blackhall, who is the most incredible thoracic oncologist who has a real deep understanding of lung cancer biology, as well as the clinical application and the treatment of patients. So she and I got together in about 2006, I think, if I remember correctly.
Okay.
I was getting the Biomarker Centre going, and she'd come back from Toronto, where she'd been doing training in cancer research, lung cancer research, not just the clinical part, the laboratory part too. So she's one of those clever people that can do everything. And she and I got together, and we were interested in, we just bought a new technology. At the time it was called cell search, and it was a technology that allowed you to find and count circulating tumour cells from a sample of a cancer patient's blood. And the very first sample that we ran through this new technology back then just happened to be one of Fiona's patients who had small cell lung cancer.
Really?
And what we discovered is, we all knew that circulating tumour cells were going to be vanishingly rare.
Yeah.
So bazillions of blood cells in a blood sample, because that's where they live, and occasionally a rare circulating tumour cell. So we were quite surprised in some of our first samples from small cell lung cancer patients that actually there were quite a large number of circulating tumour cells. So we perhaps shouldn't have been so surprised, because we know this, as you said, the most aggressive of lung cancers, it is metastatic very early.
Yeah.
So most patients who present to the clinic have already got metastases.
Yeah.
So maybe it's not such a surprise that we found quite a lot of circulating tumour cells, because that's how the tumour that starts in the lung moves around the body and moves to other sites. So maybe it wasn't such a surprise, but it was perhaps quite lucky, because we then went on to sort of count them, realised that the number of these circulating tumour cells was related to how long a patient would live.
Yeah.
We found out that the number of these circulating cells disappeared when you gave the patient their first cycle of chemotherapy, because most patients respond quite well at first.
Yeah.
And then we discovered that those circulating tumour cells went up in number again as the patient starts to become resistant to their chemotherapy, which almost all patients with small cell lung cancer do.
Yeah.
So that was the sort of basics of, circulating tumour cells are a barometer, if you will, of how a patient with small cell lung cancer is responding to their treatment. And we've got much more elegant with our studies since then. And I guess the biggest breakthrough that we made, the team with Fiona's team and our lab-based team, as we really started thinking about what more can we learn from these circulating tumour cells. And because small cell is the disease it is, it's very aggressive, and it's very hard to get tissue biopsies. It's not so much like breast cancer, where a tissue biopsy is a lot easier. These are deep-seated in the lung, and you can't really keep going deep-seated into the lung and taking a piece of tissue out. And even if you do, they're very small, and they're quite necrotic, so they're difficult for a researcher to work with. Yeah. And doing it over and over again is not really an option. No. And because we know that patients respond to their chemotherapy, and until very recently, that was all that was really available for them. Immunotherapy is making some impact, but not a huge impact. So going in and taking tissue biopsies isn't really ethical even. It's not really what you would do as a routine in small cell lung cancer. But the fact there are so many of these circulating tumour cells made us think, well, how do we get to study the biology of small cell lung cancer? How can we do that when we don't get tissue biopsies hardly ever? Yeah. So what we did is we found the circulating tumour cells. We did some experiments to remove as many of the blood cells from the blood tube that we can. So we try and enrich for as many circulating tumour cells and as few blood cells. Okay. It's not perfect, but then we take that enriched circulating tumour cell sample and we put that into a mouse, and we grow what effectively looks exactly like the patient's tumour in a mouse model that we can then study. Yeah. So one of the other things that's really, really, really challenging in the clinic and for small cell lung cancer patients is many of them get metastasis to the brain. Yeah. Well, getting a biopsy out of the brain, the human brain, really hard. So, and it's devastating. it's a really big unmet clinical need, as is the treatment of small cell across the piece. But what we can do now is we have models where we can get those brain metastases in the mouse. We can study the biology.
Yeah.
And only by understanding the biology of any cancer can you really, truly get new therapies that have a good chance of being effective. So I think the landmark sort of change for us was understanding that there were lots of circulating tumour cells compared with any other cancer type that we'd studied, and that we could get more out of this finding than just counting them.
Yeah.
We could actually make new models with them and really begin to much better understand the biology of this really aggressive disease. Yeah. And what I'm really pleased to say is, this methodology that we develop, these models that we call CDX, everybody thinks they stand for Caroline Dive xenografts. They don't. They stand for circulating tumour cell patient explants. Okay. But anyway, that's beside the point. But this new methodology for making these CDX models from a patient's circulating tumour cells is now being developed across the world. So I was really proud of that discovery. I think the team that put all of that together wasn't just me, it was a big team. Yeah. I think we have made a step change in understanding the study of small cell lung cancer. Yeah. And we have, I think, some of the unique brain metastasis models now, and we're very, very busy in the lab trying to understand what does a tumour cell have to do to get to the brain, to stay there, to survive there, and then to grow there. Yeah. So what changes in the biology have to happen for a brain metastasis to grow? And it's absolutely fascinating. I won't talk about it in detail today, but we're beginning to understand what those processes are. And, and the hope will be that one day we'll understand it well enough to figure out how to slow it down or stop it.
Well, you say that. I was thinking this before our talk. I was like, what can I talk to Caroline Dive about that will fit within an hour slot? And it's, you've got to really blooming pick. And we've chosen to focus on lung cancer, but maybe it'll be something else on another episode. But
well, there's more, actually, because, the big discovery in small cell lung cancer worldwide is when we think about, let me change tack a bit. When we think about breast cancer or even non-small cell lung cancer, if you think of a pie chart, we can say that a patient's tumour fits somewhere in a segment of that pie chart. Yeah. So there are different types of breast cancer. There are different types of non-small cell lung cancer. But we just had one pie chart for small cell without any segments. It was just small cell and everybody got chemotherapy. Yeah. Like I say, some patients were responding to immunotherapy, but not the vast majority of small cell lung cancer patients. So we had no segments of the pie. Yeah. And we do now. So just recently, the whole field, it's a very tight-knit group across the world that study small cell lung cancer because it's a really difficult topic. And we've all got together and collectively we've come up with a new classification of small cell lung cancer, which was published in Nature reviews recently. But they're based on what's called transcription factors. So not based on mutations, based on transcription factors. And there are at least 4 or maybe 5 subtypes. Yeah. And the hope is, and the studies are beginning to suggest, that different subtypes will respond to different types of drug differently. Yeah. So now there's a chance that we could stratify or pick the right patient for the right drug for the first time in small cell lung cancer. We're not there yet, but it's coming. For the first time in 30 decades, there's been a step change in how we think about the treatment of small cell lung cancer. So what we decided to think about then was, we need a blood test because of all the problems with getting tissue biopsies. We need a blood test that tells us what subtype they are. So the very recent publication from our group, we've managed to do that from the blood. So and this is going back now, not to the circulating tumour cells, but going back to the ctDNA. Yeah. So what we can do now is not just measure, is it there, is it not there, or what mutations are present. We can actually measure molecules which alter the profile of that circulating tumour DNA. So it's called methylation. Yeah. So these are putting methylation groups or molecules onto DNA that changes how we recognise that DNA. And that's important for lots of reasons. One is it tells us where in the body that circulating tumour DNA comes from, in this case from the lung. But more than that, we now have developed these profiles of the circulating tumour DNA that tell us which subtype it is. Okay. So working with pharma, working with biotech, new trials will be coming forward where when we've fully validated this test, so we've just started on a journey with this. So we've got lots of new samples coming in from around the world from clinical trials in small cell lung cancer where we now can test our test. Yeah. And if that works out, the next step will be a new clinical trial going forward where we take the blood, we say it's this particular subtype, and therefore this might be the best drug for that patient for the first time in the history of small cell lung cancer. So this is not just my group, obviously, not my biomarker centre. This is a global effort.
Yeah.
But I like to think we've made an important step forward in it.
You really blooming have. And just a kind of simplification of that. So we've got our circulating tumour DNA, which is just DNA, the instruction manual of a tumour cell. And within that, we've got the bases, which is A, G, C, and T. I don't know how far people go back. You might have learned
biology
that.
A-level.
GCSE biology. And the methyl groups that you mentioned, so they will attach to the DNA.
Yes, so that's they instruct gene expression. So whether a gene is on or off. Yeah. And whether a gene is on or off tells you a little bit about which tissue it's in.
Okay.
So we can use this in lots of ways. So I've been telling you about how we might subtype small cell lung cancer, but there's another tumour type where this is going to be really, I think, important.
Oh, go on.
And that's called cancer of unknown primary. Yeah. So we're working with our colleagues in the Christie, Natalie Cook is leading from the clinic, and we trained a clinical fellow in the lab called Alicia Conway, and together with the team in the Cancer Biomarker Centre, Dominic Rothwell, Alex Clipson, Simon Pearce, lots of people with lots of different skills came together to develop a new test, and that is to use this circulating free DNA methylation that we were talking about to find out where, so the patient presents with a metastasis, but no one knows where the original tumour came from. Yeah. And to know how to treat the metastasis, it's really important to know where the original tumour came from. And now, because these methylation on cfDNA give us a postcode, we can infer where that metastasis came from. Yeah. So we can say it was a colon, or it was a prostate, or it was a breast. Yeah. And of course, once you know that, and you know the mutation landscape through the mutations in the circulating tumour DNA, you can now say, well, if this was a cancer of unknown primary from the colon with this particular mutation, the best therapy would be. Yes, and it's that approach that we've developed as well. So the nice thing about becoming sort of focused on liquid biopsies is you start to think about all the different clinical utilities. Yeah. So whilst we focus on small cell lung cancer biology and liquid biopsies for small cell lung cancer treatment, what we learned there helped us develop a different test for a different clinical question. Yeah. So we try and go for the really difficult clinical questions that really need an answer.
As usual. Yeah, it's amazing. And I think looking into small cell lung cancer as that thing that was treated with platinum-based chemotherapy. And like you say, you know, the prognosis for it has been poor in the past, and it's looking to the future and looking at specialising, because as soon as you know the subtypes of a type of cancer, your treatment changes, and that is the precision medicine.
That's the anticipation. Yeah, we have to do the experiments clinically and preclinically to get there. But I think it's hope where there wasn't very much for the last 30 years that treatment will change for small cell.
For sure. And just looking back at the CDX models, so that was in 2014, was it?
That's when we first started, the first eureka moment where a tumour grew. We were just so excited that we developed these new models.
And you were the first group ever to do that?
We were the first group to do it in small cell lung cancer. There had been a group in Germany that had done it for breast cancer, but they had made hundreds of attempts to make one model, because the circulating tumour cell count in breast cancer is much lower. Yeah. And those cells don't appear to be quite as tumorigenic in the mouse models as the small cell lung cancer. So we can go down to just 5 cells and put those back into the mouse, and they'll grow a tumour. So they are really, really aggressive in small cell.
And is it higher in small cell because of the high metastatic rate?
I think that's the clear indication. I think, you know, when we think about these cells, they're neuroendocrine, and they undergo quite a lot of plasticity changes. And what I mean by that is, if you think about caterpillars and butterflies, their genes don't change, their mutations don't. Well, they don't have mutations, but nothing changes at the DNA level. But the post-DNA level, everything changes, and the caterpillar looks nothing like a butterfly. Yeah. And we think the small cell lung cancer cells are very good at going from caterpillars to butterflies, if you will. They can mimic, for example, one of our other discoveries is that they can mimic endothelial cells. Okay. So they start off as neuroendocrine, so a certain type of cell, and then they can undergo a switch to non-neuroendocrine. And when they're non-neuroendocrine, they behave like the cells that make blood vessels. Right. So we think they can make their own vessels inside the tumour. to get nutrients and oxygen to help the tumour grow. And that's an example of, I would call, functional plasticity.
Yeah.
So that's the tumour cell mimicking another cell type
Yeah.
for a particular function.
Yeah.
That's to the benefit of the tumour.
Yeah.
So we're pretty excited about that, you know. But these small cell lung cancers, this is why it's difficult to treat, because it's a moving target.
Yeah, exactly.
And we have to think of combinations. So what would be the drug that would treat the cell when it's neuroendocrine? It might be a different therapy when it's switched to non-neuroendocrine.
Yeah.
And if the tumour's composed of both of those phenotypes, then we might need a combination therapy that hits both.
Yeah.
You know, so there's lots to think about. Once you start understanding the biology, you can start thinking about how do you treat this tumour.
Yeah.
And that's very much, I think, completely important, because, you know, the CRUK Manchester Institute does really fundamental biology research, cancer biology research, as well as the Biomarker Centre that's much more translational, sitting in between that basic science and the clinic. And that's where we come back to the new building.
Yeah.
Because that's the synergy that we now have in that new building between the folks doing curiosity-driven, fundamental, basic science.
Yeah.
Someone like myself that sits in the middle on the bridge to the clinicians.
Yeah.
And it's that backwards and forwards between clinical, translational and basic research going in both directions that I think generates progress.
Yeah. Bench to bedside.
That's exactly it.
And then bedside to bench.
Both directions, for sure.
Could you just explain a little about what a neuroendocrine cell is?
It depends on where the cell originated. So for small cell lung cancer, the main cell of origin is called a neuroendocrine cell body in the lung. So that's a specialist type of cell that's in a normal lung. And it's there where the first, if you will, mutagenic events take place that drive small cell lung cancer.
Okay, interesting, cool. And then just CTCs again. Sorry, I really
We're
want
jumping around, but that's
yeah,
fine.
no, we are jumping, but it's fine. The case study that I'd like to focus on is CTCs post-tumor resection. So, did you do a paper? Was it on small cell lung cancer where you monitored CTCs, so the circulating tumour cells post resection of the tumour?
Yeah,
in
so
the
that
lung?
was it. That was in the preclinical model.
Okay.
So that's how we ended up trying. It's how we've got to the point where we can really study the brain metastasis.
Okay.
So you can grow the tumour, and then the tumour gets to a certain size. You can take it off the mouse. The mouse stays alive, but that mouse already has seen circulating tumour cells move around, and you wait, and then that mouse may develop a brain metastasis or a liver metastasis.
Okay.
That gives you a really interesting question that you can start to study, which is, what is it special about the tumour cells that go to the brain, and are they different from the tumour cells that go to the liver?
Yeah.
And is the adaptation to the brain environment clearly different from the environment of the liver? So what do those tumour cells have to do to grow in the brain that's different from what they have to do to grow in the liver? So again, all of those questions are hard to address. So what we do is we have our preclinical models that we can study and do function testing experiments in. So one of our subtype molecules we can knock out in the mouse model and say, do those cells still go to the liver? And the answer is no. So that says that very important molecule has a role to play in liver metastasis. So there's lots of ways where we can have the preclinical models and the collection of clinical samples, and we go backwards and forwards between the preclinical model and the clinical samples. Is what we see in the preclinical model relevant to what's happening in a patient? Because that's what we're striving for.
Yeah.
You know, and what we see in the patient samples, there's a brilliant protocol nationwide run from the CRUK and UCL within the Lung Cancer Centre of Excellence with us in Manchester by Charlie Swanton and Mariam Al-Hajani, working with Fiona Blackall and myself, called Peace.
Okay.
And there, when folks sadly pass away, they've given permission for warm autopsy or for an autopsy.
Yeah.
And that's the only chance we've got of getting a brain metastasis from a human being to study. But does that brain metastasis from a human being look like the brain metastasis that we've developed in our model? So it's a way of validating the preclinical models to make sure they're patient relevant.
It really is incredible stuff. And you think about, 2014, that the inception of this in small cell lung cancer. And then I guess, is it a signifier for prognosis of that, the high levels of CTCs post-resection, and maybe if you've got a brain and liver metastasis, I guess.
Well, we don't know that, because most patients who have small cell lung cancer don't have surgery.
Okay.
Because when they're diagnosed, it's too late for surgery.
Yeah.
It's already metastasized.
Yeah.
So that's why, and again, we don't get very often resected material from a patient to study.
Yeah.
So that's the challenge, and that's why these circulating tumour cell models are, I think, a step change in understanding that biology that we wouldn't otherwise have the opportunity to study. We can put tumour cells in plastic dishes and we can grow them in the lab, but the plastic dish doesn't really look much like the patient. So, we can do certain things with cell lines in plastic dishes, but I think we wanted to have a more elegant model than that.
Cool. It's amazing. So, I mean, you've talked about the use of biomarkers here. So we've specifically focused on CTCs, CF or tDNA. And I'm wondering, what are the current barriers that you envisage to getting those liquid biopsies into the clinic and used in a more routine manner that could be, for instance, a patient having early detection, or the tracking of their prognosis, the tracking of how their treatment is going? What do you envisage as the barriers to...
I mean, it's quite a hard and long road because, as I said, you have to test the test. So you have to design really good clinical trials to test the test, and you have to show that the test works, because otherwise you can't. Why would you use a test that you're not sure about routinely for cancer patients? So it's a bit like drug discovery. You start off with lots of possibilities and you end up with very few that make it all the way to the clinic. Because it's a difficult road. And the same is true of these liquid biopsies. We have to validate them and we have to make sure that they're accurate. So those can sometimes take years to do the early detection experiment. So there are many companies around the globe, including Grail and the Galleri test in the NHS, thousands and thousands of participants in that experiment to find out, does that test detect cancer early? How many cancers types does it detect early? And how early does it detect them? And how often does it detect them early? All of those questions will come out of that huge experiment. But it will take a lot of money and a lot of time and a lot of people participating to get there.
And I think we've spoken to people before that have got their, research into clinical practice or changed guidelines and things, and the actual process behind that is arduous and so convoluted, in the sense that if you're, working with NICE or NIHR to implement in the NHS, you need your evidence basis, you need your economic modelling, and things such as that take years.
Not my areas of expertise at all.
They wouldn't be, because you've got loads of other areas of expertise. But it's
exactly. But that's the route. There is a roadmap. I mean, we know what it is, but it's a hard, long road. And I think we just have to keep plugging away until we get to the point where we're confident and we've proven with evidence that the test works.
For sure. And I mean, what are your kind of future hopes of the CBC? Like, what is this roadmap that you speak of?
Well, I think we have about 100 staff at the moment. And what I'd like to see in the coming 5-10 years is that we'll be more popular. So we want to increase the number of folks that we've trained to be specialists in cancer biomarker sciences. We want to think a little bit more about a wider scope of types of molecule that we measure. We haven't even thought about the microbiome, so the influence of bacteria and so forth on tumour biology. And there's a new junior group leader at the Institute who's beginning to get into that space. So the basic science has to give us enough information to understand how we might develop biomarkers in that space. And I think that will be very challenging. There's a whole new area of cancer metabolism. So how cancer cells change their metabolism to their own advantage to grow and to invade and to metastasize. And I think there will be biomarkers in that space that haven't been developed for clinic yet. And that's something we would like to expand into as the basic science evolves and we get more information, so that we can really, we don't want to be plucking biomarkers out of the air. We want them to be evidence-based. So that's another area. And I think what we want to do is increase the number of collaborations that we do. So we're very collaborative already. I think I can't even tell you how many clinicians at the Christie we've worked with over the years. But we'd like to work with more folks with different areas of expertise and really expand our scope. So capacity and scope, new areas for biomarker development. And it's all predicated on what is needed in the clinic. And that, again, is why having a biomarker centre at the same place as the Christie Hospital is ideal, because we have that constant connection with the clinic. So more and better, I guess, is the simple answer to your question.
Yeah.
But listening to the clinical needs, listening to our clinical colleagues, what do they need? What clinical questions are the most important clinical questions that need biomarker solutions?
Yeah.
And being much more nationally engaged. So we work with most of the Cancer Research UK experimental cancer medicine centres.
Yeah.
But again, to really be fundamentally working across that network across the country as well.
Yeah. And we've mentioned collaborations there, and the one that's come to mind is TracerX, because it's been in the news quite a bit, and I know you work very closely with Charlie Swanton. Yeah. Could you tell me a bit about the small cell cancer work that you did within that TracerX consortium?
We worked with Charlie on non-small cell lung cancer predominantly, although the Lung Cancer Centre of Excellence that he and I direct between London and Manchester does of all types of lung cancer. But in TracerX, we sort of used our expertise in circulating tumour cells, and I think one of the studies we did together with Charlie's teams, and a huge national effort really, was we asked whether could you look at circulating tumour cells in early stage disease? Mm-hmm. Were they there? So everyone's, in the past, there's sort of been a thought that metastasis was a late event.
Yeah.
But actually, it's quite clear now that circulating tumour cells are out and about quite early.
Yeah.
So even in the earliest stages of non-small cell lung cancer. And rather than taking the blood sample from the arm, where we didn't find very many, we took it from the pulmonary draining vein of when patients were having their tumour resected from the lung. Okay. So this is, I think, what you were referring to before.
Yeah, I was.
So when a patient has a tumour resected, just before the tumour comes out of the lung, when the surgeon's busy, we take some blood from the pulmonary draining vein. So that's the lung that drains the cancerous lung. It's the vein that drains the cancerous lung. Yeah. And we looked for circulating tumour cells there, and we found them. Okay. And when we found them, the number that we found seemed to predict whether that patient's tumour after resection would come back and what their survival would look like. So we think the number of circulating tumour cells early is a predictor of relapse. Okay. So that's the work we did in TracerX, and we're going to do more of it in TracerX Evo, which is the next big study inside the Lung Cancer Centre of Excellence between Manchester and London. And again, we want to now test the test. Yeah. So is that number of circulating tumour cells early at resection really predicting, with really solid evidence in many more patients, does it predict relapse? But we did a bit more than that. So each single cell that we found in that pulmonary draining vein, we sequenced. And what was really interesting is some of them didn't look like cancer at all. So when we looked at their molecular profiles, they just looked normal. So they're definitely epithelial cells. They're not blood cells, but they're in the blood, which is odd. Yeah. But they don't look like tumour. Yeah. So we don't know what they are yet, and that's something else we want to find out. But the number of those and the ones that do look like tumour cells, that combined number predicts relapse. Okay. And then the other thing we did in a couple of patients is we looked at all the mutations in each individual circulating tumour cell at that point that the tumour was resected. Yeah. And we looked at mutation profile. And what Charlie's team did was the resected tumour was cut into many pieces, and they looked at the mutations in the tumour, the original tumour. And then when a patient got a metastasis, in this case 10 months later, we compared the mutations we found in the circulating tumour cells, the mutations that Charlie found and his team found in the resected tumour, and we said, well, what does the metastasis look like from that mutation profile? And what we found is that there were lots of mutations in the circulating tumour cells that we hadn't found in the original tumour because they were so rare, but they appear in the metastasis. So putting all of that together, it sort of says that these early circulating tumour cells are the architects, if you will, of the metastasis that comes later. Yeah. So that springs another question. What were they doing all that time? Yeah. They were presumably just sitting, not proliferating, and at some point they proliferate. So there's lots of work that we need to do now in the next version of TracerX. There's a huge study going on between London and Manchester, which is going to be amazingly exciting, even more brilliant science coming out. So I think, when Charlie and I started the Lung Cancer Centre of Excellence, more than 10 years ago now, there just weren't very many people studying lung cancer. It's difficult to study, compared with breast cancer, for example. And I'm not saying one is more important than the other. I'm just saying one is slightly easier to study. Yeah. And we felt because it was such a major killer, lung cancer, and so prevalent, particularly here in the Northwest, actually, in areas of social deprivation and so on, there just wasn't enough lung cancer research happening in the UK. And one of the things we wanted to achieve with the Lung Cancer Centre was just to train more young, bright people to do lung cancer research. And we've just a few weeks ago come back from York. So every other year, excepting COVID, we take all the young researchers up to York and we have a couple of days where we all talk about our science together, and it's absolutely brilliant. And it's just wonderful for me to reflect on that over the past decade of how many young scientists we've trained in lung cancer research who are all doing amazing things now. So and the number of students doing lung cancer research has really grown over the period of the Lung Cancer Centre of Excellence. And of all the things I do, I like training young bright things best. So that's particularly pleasing for me.
Yeah, like sponges. And I guess you've mentioned as well, because I had conversations with Phil Crosby in season 1, and he was talking as well about the lack of research behind lung cancer. And there's also that stigmatization factor that breast was always funded really well and you'd get a lot of breast cancer research funding. So it obviously kind of...
Yeah, and I think, what TracerX and the Lung Centre have done is balanced that up a little bit.
Exactly.
But all cancer research is important.
Yeah, exactly.
When you think of young women who get breast cancer, we certainly don't want to downplay that in any way.
No.
But an enormous amount of people have lung cancer, and we just didn't really have enough effort in that area, which I think would be, with TracerX and TracerX Evo, we're now beginning to rectify. We have rectified, I think, in this country.
For sure,
but the next challenge is never smoker lung cancer, and many people have heard Charlie Swanton talk about that, and that is another puzzle to solve.
Yes, we actually had Fabio Gomez on the episode before, so we were talking about ALK positive, never smoking, and like you say, it's that education piece, isn't it? And I think, like you say, the more research that you do, the more research questions you develop. So it's like catch-22.
Well, and the more biology you understand, the more rational use of therapy you can think about. So it's been an extraordinary time here in Manchester. I don't regret a single minute of it. I mean, as I say, I'm a Sussex girl, but to do the work we do, this is the place.
Yeah. How many years do you think you've got left? 40?
Oh, that's a horrible question.
40 years, 50 years?
Come on. No. I think for me, what I'd like to think towards the end of my career down the line is, what do you leave behind? And did you do something meaningful?
Yeah.
And what I'd like to see is that Cancer Biomarker Centre go from strength to strength.
Yeah.
There's every reason why it should. And that would be, I could look back on that and be pretty proud.
Your Cancer Biomarker Centre. No, I'm looking forward to... seeing that to fruition and seeing what becomes of it, because there's so many exciting things that I think are going to come from that, and you've laid the groundwork so well.
Thank you.
And like you say, it is in such a great position. We've got the Christie for all this patient samples and things, and the team there are just phenomenal. Like, even from walking around, I was absolutely blown away.
Well, I mean, I also like bringing the clinical fellows. So we train folks who've come through a sort of a basic science route, undergraduate degrees, but I also really enjoy training the clinical fellows that come through.
Yeah.
And often they come into the lab, they've never held a pipette before.
Yeah.
But they're very smart people.
Yeah.
It doesn't take them long to catch on. And it's really nice that what we can learn from them, what they can learn from us. And it's that different languages, different training, but a common goal. Yeah. And that's what really works, I think.
And and like you've said, understanding the clinical needs. So all your research questions are informed by that clinical need and the questions that clinicians are asking. It's amazing stuff. And honestly, I think, I've got a bit of a background in science, and reading about all this and doing my research kind of preemptively before this, I was just blown away by all the stuff that you guys do. And I think it's also understanding that even though things haven't seen fruition in terms of being applied in that clinical setting, and, you've not got the biomarkers in a GP routine setting where somebody has a blood sample.
It's coming.
It's coming. You look at the hope of things and you understand there is a lot behind it. There's that clinical trial need and the validation of, the tests, like you've said, again and again and again. But it's the hope of understanding that things are looking really good.
Yes, and even in the phase 1 setting, which, where patients have sort of failed available therapies to, and they go into this experiment that's called the phase 1 trial, where a new therapy that's never been in a human before, they gradually increase the dose through folks coming through the phase 1 unit to find the right dose, to make sure it's safe. We're learning more even in that setting. So I don't know whether you've spoken to Matthew Krebs, who led the Target trial in the phase 1 unit. Now that's nice, because Matt was one of those clinical fellows that did their PhD with us and is now running what's called Target National. But what we collectively did there was we actually started to look at that circulating tumour DNA profiling in the folks that are being selected for a phase 1 clinical trial. And whereas that was a little bit more empiric in the past, we can now say, well, this is the right clinical trial. This is the clinical trial for you that has the best chance of giving a benefit, because we understand your molecular profile. Not only we've had your tumour sequence, but we've also sequenced your circulating tumour DNA, and you put those data sets together. And that's helping the clinicians, in this case, Matt Krebs, who's running that trial, help pick the best trial for you. And it's not every patient that will benefit from that approach, but certainly some will. And that whole process, as well as the virtual molecular tumour board, which is the sort of the dry science that we do in the Biomarker Centre by our digital team. We made, we couldn't all get together in COVID to discuss all the data, but we had already developed a molecular tumour board that you could all have on your computer screen, didn't matter where you were sitting, and you could all look at that data in real time together and discuss it. So that's called eTarget. Well, that's now been adopted across the country in Target National. So, some of the work that started off, will you come, Nick Jones said, come on a Friday and do pharmacokinetics on a Friday afternoon. It's sort of, at this point, culminated in a national phase 1 clinical trial called Target National. So, it's been a journey along the way, but things are happening. So,
and there's a lot of things to be proud of there. I mean, you must love looking back, in the same sense that you love looking forward, but there's so many remarkable...
It just makes me feel a bit old.
No. You've just crammed a lot into a few years.
A long time. But, lots of new folks have come and joined us in Manchester, and this whole team science approach, which I know you spoke to Rob Brister about. It's something Manchester's really good at. And that's why I'm here. I enjoy that collaborative spirit that we have here in Manchester.
Oh, it's great. Honestly, I think that's why I'm here too. You love speaking to somebody, asking if they know somebody, and they're like, I did this paper with them, and you think, what the heck? But it is just amazing.
And a shout out to the Christie. Over the years, you begin to realise very quickly what an incredible place the Christie is. I've heard so many people tell me how they've had relatives who've been through the Christie and what wonderful care they got. So we are really privileged to have the science and that Christie Hospital right here.
I love that as well. It's a hard thing when you hear that somebody's had a cancer diagnosis, but I breathe a sigh of relief when somebody tells me they're being treated in the Christie.
Sure.
And
absolutely.
I sent an email really recently, and I just said, look, you're in the best hands possible. But anyway, just a question. What's your favourite biomarker, Caroline?
My favourite? Oh, goodness, I don't know. I think my favourite biomarker ultimately will be the one that actually makes a difference in the clinic. So I think this blood test we're developing for cancer of unknown primary, I think we've got a lot of work to do, but I think that has a real opportunity because, patients who come in with, we call it CUP, what my clinical colleagues tell me is it can take almost a year sometimes to get a diagnosis. Whereas I think in 2 or 3 weeks with the blood test, if it works as well as I think it's going to, and that needs evidence. But if that comes to pass, you could be giving them a diagnosis or at least informing your clinical colleague who's looking after that patient, this looks like it's come from whichever organ, the breast, the prostate, the colon, whatever, and give them information that really helps them make a treatment decision for that patient in a much quicker way than the current practice. So we will do an enormous amount of work to get that through. But I think I can see the potential for that blood test. If we get to a point where we can subtype small cell lung cancer with a liquid biopsy, put folks onto to targeted therapies that have the best chance of giving them a good effect, that will also be something that I think will be incredible.
Fab. So just to summarise our discussion, I guess these biomarkers are detection agents, subtyping agents, and looking at the prognosis and relapse potential of a cancer.
Response, how quickly is a patient responding, when to switch therapy, because the tumour is evolving and becoming resistant. When is the tumour coming back after surgery? I mean, there are lots and lots of ways that biomarkers can help support patients. And in our biomedical research centre in Manchester, which was a huge effort by so many people to have that increased in scope and amount of funding in Manchester just recently. We're in year 1 now of BRC2. There are 4 cancer themes. There's cancer precision medicine that I lead on. There's early detection, Emma Crosby, that advanced radiotherapy, and living with and beyond cancer, which was the new one that John Radford and Kim Linton run. And, actually the journey for someone who's had cancer, it is a long one, and if you might get a secondary cancer, you might have cancers come back, which are years and years after your treatment. So having biomarkers that you can use in the longer term to live with and beyond cancer, I think that's a really important area, and I think John and Kim really are developing that theme really well, and we're obviously playing some role in that one as well. So biomarkers really can be used right across the entire spectrum of cancer patients, from early detection all the way to living with and beyond. So we've got plenty to keep us occupied. That's for certain. Sure.
And let's not forget that it completely changes the invasiveness of that investigation as well. Because some people might not like blood tests, but they are certainly a lot less invasive than a resection or a surgery.
And I think, we will still need tissue biopsies.
Yeah.
We will still need to know that what we're measuring in the blood is relevant to what's happening in the patient's tumour. And an area where it's particularly difficult is immunotherapy.
Yeah.
So one of the things that's a big goal going forward is how do we develop predictive biomarkers to tell who's going to benefit from immunotherapy, the reawakening of a patient's immune system to tackle their cancer. And there's been some incredible developments, as you know, in oncology with immunotherapy. And some patients get remarkable responses that are really durable, and others don't.
Yeah.
And we don't really know yet how to pick the winners, so and who is not going to respond. And we don't want to give expensive and sometimes toxic therapy to patients who are not going to respond.
Yeah.
So it's that predictive biomarker for immunotherapy, which is a huge ambition and goal that we must tackle.
Yeah.
So whether we'll need a tissue biopsy or whether we can do that in a blood test remains to be seen. But the tissue biopsy obviously gives you the whole microenvironment, including all the immune landscape inside a patient's tumour. And that we're going to need to really understand if we're going to develop biomarkers.
Great stuff. We'll be talking again, Caroline.
Okay. Give us a little while to fix that one.
Yeah. So my 2 final questions are quite personal, but I guess it's just, you talked about your granddad and that inspiration behind becoming involved in cancer research. How does it feel to know that, you're benefiting such a vast patient population?
Well, cancer research, there's very few of us that end up with, few folks who end up with a Nobel Prize. The guy that understood immunology enough to think about these immunotherapy agents, just being one clear example. The guy that figured out how cells divide being another. Folks who did the structure of DNA. We're not all going to end up with Nobel Prizes, that's for sure. So everyone makes their contribution in small iterative steps.
Yeah.
I actually think training the next generation is one of the ways I've perhaps made the best contribution.
Yeah.
And I'd like to continue to do that. But I think, looking back on a career, you just want to make sure you gave it all you had.
Yeah.
I think that's I think the thing that drives me nuts is laziness and boredom. Are my 2 most hated things. And if I ever feel them in myself, I get very, very upset. So I think, it's just giving it everything, giving it your very best shot.
Yeah, for sure.
And that's what a career in cancer research should be about, I think. So, we're not all going to win Nobel Prizes, but we'll all make a contribution in our own ways. And for me, training the next generation is actually really important.
Yeah, for sure. Teachers are the most valuable people. Because otherwise, how are you going to have a succession to anything?
And that life is ironic, isn't it? Because my own father died last December of cancer of unknown primary in his liver.
Crikey.
So I was a bit fed up about that for all sorts of reasons, obviously, but I wish I'd just got my blood test done a bit quicker.
Yeah.
But it's just, there's an irony there,
know.
But bless him. He drove discipline into me and made me who I am, so good on him.
Caroline, you couldn't have done it any quicker, I'm sure. You're very, very steadfast in the way that you move. But I'm sure he'd be absolutely blown away by what you're doing now. And I guess my final question is, how do you do it all? I mean, you've already said, you don't like boredom or laziness. So I'm kind of anticipating that you have a very fast-paced life.
Yeah, I do everything quite quickly, I suppose.
How do you do it all?
Everyone asks that question. It's quite a difficult one. I just do. I don't know quite how to I don't know how to answer it. I did have a very disciplined childhood. My dad was in the army for a long time, so there was never any unstructured time in my childhood. And I guess that I've learned important lessons of how to be self-disciplined and organised. Although if you ask my husband, he would just laugh because I'm very disorganised at home. But no, I love being busy, and I don't like time wasting. So I'm either busy on the tennis court, or I'm busy at work, or I like to travel. I don't often sit down and do nothing. Because I just don't enjoy sitting down and doing nothing. So I don't know. I guess the default position is busy.
Yeah. No, I'm the same. I have to occupy my brain at all times. It has to be occupied. I have to be doing something. And some people aren't like that. But if you are, it becomes the par for the course. You have to do things.
I think I quite like problem solving.
Yeah.
So my daughter always tells me, Mum, you don't have to solve every problem. But
yes, I do.
It's kind of who I am. So I do like to solve problems.
Yeah. And I mean, it's an exciting legacy that you have at the minute, and it will be very exciting in the next 5-10 years as well, which is...
Watch this space,
Watch this space.
Indeed.
Well, honestly, thank you so much. I know it's difficult getting time in your diary, and I really appreciate the time that we've had with you today, and I hope people have really enjoyed. For anything that we've mentioned, I'll link in the show notes the papers, Tracer X, and also just go and have a bit of a read about the Biomarker Centre, because there's some exciting things that are going on there for sure. But honestly, thank you for your time, your input, your wise words, and a bit of a background about you. It's been really great to dive into the world of Caroline Dive.
Dive into Dive. Okay.
Thank
that
you
one
so
before.
Much. No, it's original.
Thanks. It's been great to talk to
Thank
you,
you
Sally.
so much. And we'll see you next time. Bye.
Okay. Goodbye now.
If you have been affected by anything you've heard in this episode, please see the show notes for a list of charities and organisations that can help.
1 in 2 was brought to you by the University of Manchester and the Manchester Cancer Research Centre. Listen to our next episode to hear from more of our researchers as they share the innovations, discoveries and projects that are changing the landscape of cancer prevention, early detection and treatment. Heard today, please see the show notes for this episode, where you'll find a transcript and links to further information and research. Cancer is one of the university's 5 research beacons, showcasing the interdisciplinary collaborations and cross-sector partnerships that are tackling some of the biggest questions facing the planet. To hear more about Manchester's research in advanced materials, biotechnology, cancer, energy and global inequalities, go to manchester.ac.uk/beacons.